Good morning, everybody. My name is Maciej Czerwinski. I'm a director of consulting at XenoTech. Today, I would like to tell you a little bit about our biobank, fatty liver disease tissues we have in that bank, and the research applications. First, I'd like to say that XenoTech has an awesome on-call team of talented individuals that specialize in isolating human hepatocytes. Over the years, many of my colleagues work on the team and processed so far close to 1,500 human livers. This large number of livers represents variability of the donor population in the US. In addition to hepatocytes used in our regulated studies, such as enzyme induction studies, we produce subcellular fractions from the organs we receive. These fractions support a range of biochemical, protein, and nucleic acid studies. But the data obtained in these studies are only a part of what we can learn from the tissues, since when cells are separated and isolated, their relative spatial distribution of organelles or molecules present in the liver is not fully appreciated. This information is lost in isolated cells or isolated subcellular fractions. Hepatocyte studies in the presence of their neighboring cells can reveal more that is viewed in the isolation. Our biobank was therefore established to preserve, characterize histologically, and distribute a diverse selection of samples of normal and diseased organs. Obviously, fatty liver disease is the focus of our collection, with the major categories being normal livers, necessary for a proper baseline or a background, samples with steatosis, and samples with steatohepatitis, either with a history of alcohol consumption or without, which would be a NASH. From the samples gathered in the biobank, we provide lysate, subcellular fractions, and microarrays. I underline microarrays because this is the research tool that allows for utilization of spatial data and some analysis that is not possible with isolated cells or subcellular fractions. Tissues in the bank are described by their demographic data, serology, and information on donors' health, such as BMI, body mass index, alcohol use, diabetes, and abundance of macrovesicular fat in the liver. Serology for HIV, hepatitis B, and hepatitis C are always negative. These are our acceptance criteria. Percent of macrovesicular fat is estimated from hematoxylin and eosin slides, which are routinely prepared for tissues that are considered or accepted into the biobank. In the first image, I'll share with you view of a normal liver. At first glance, we recognize steatosis in this sample. Fat droplets are bigger than typical hepatocyte's nucleus. Therefore, this is a macrovesicular steatosis. And a good example of a microsteatotic hepatocyte is indicated by a blue arrow, and there are multiple examples of that on that slide. Some microvesicular fat is also present. Overall, fat abundance is under 5% in this specimen, and the 5% is a cutoff value distinguishing normal from steatotic liver. A low magnification image is usually used for reliable estimation of fat content, as the fat droplets are not often distributed evenly. The concentration is different in different parts of the lobule or different parts of the organ. Black arrows here point to the central region, and red arrow points to the periportal tract. Within a lobule, hepatocytes are arranged in zones 1, which is close to the periportal, 2, somewhere farther away from it, and 3, that is close to the central. And these are named according to the direction of the blood flow from periportal to the central region. In the next slide, we will see details of the periportal region from the same normal donor tissue. This higher magnification illustrates additional features of the normal tissue. We observe here mild cellular proliferation in the portal region, and black arrows point out to the increased number of inflammatory cells, and the red arrows bring our attention to uneven lines separating portal area and the hepatocytes. Ideally, or in a good health, good liver health, lines separating periportal region and that of adjacent hepatocytes is smooth. Here you see a bump in that line, since there is an extra accumulation of inflammatory cells in this portal response. On this slide, presence of macrovesicular fat is also noticed as before. Inflammatory cells and Kupffer cells are present in the sinusoidal spaces, and blue arrows indicate presence of the Kupffer cells in the sinusoids. With increased accumulation of fat on a background of a picture like this, a fatty liver disease may develop. Initially, it is just steatosis, but it can progress to steatohepatitis, which would be extra fat plus an inflammation. The next slide is of liver from 39-year-old male Caucasian with BMI of 62.7, macrovesicular fat of 25%, no diabetes history or history of alcohol use. BMI of 62 is very high. Macrovesicular content of 25 is considered moderate. The BMI and macrovesicular fat content are not strongly correlated. As you can imagine, other parts of the body than the liver may be burdened with the fat load. The microvesicular fat is also present. Since we have an individual without history of alcohol use, we can think about non-alcoholic steatohepatitis. Diagnostic criteria for NASH, non-alcoholic steatohepatitis, include the following three. Macrovesicular steatosis above 5%. We certainly have it here, going up to 25%. Lobular inflammation and ballooning necrosis. Lobular inflammation is evidenced on this slide by small clusters of inflammatory cells lodged among hepatocytes, but not in the sinusoids. Balloon hepatocytes are 2 to 3 times size of normal cells. Their cytoplasm is degraded, staining lighter than in a live or normal cells, and sometimes is described as a wispy cytoplasm. Nucleus in the ballooning hepatocytes is necrotic, undergoes disintegration, and is located in the center of the cell, rather than pushed by the vacuole of fat to the side of the cell. Inflammatory cells implicated in this form of necrosis are often seen in the immediate vicinity of balloon hepatocytes. Although this slide is not stained with a collagen-specific stain, such as trichrome, fibrosis is recognized. Fibroblasts have a wavy elongated shape, also with wavy elongated nuclei. They state here on the top right. They are present in a fibrosis outside the portal regions, which are the normal localization. The next slide presents perivenular and sinusoidal fibrosis. Perivenular fibrosis is characterized by increased presence of fibroblasts around central venu, and we see central venu on the slightly right to the center of the slide, surrounded by cells staining blue. These are fibroblasts, and their collagen fibers are stained with the Mason's trichrome stain. Fibroblasts that traversing the hepatocytes in zones, in this case 3 and 2, represent sinusoidal fibrosis. You see that individual hepatocytes, regardless of their individual health status, are surrounded by elongated fibroblasts or bands of collagen. This type of fibrosis is also called chicken wire fibrosis. On this slide, you will also recognize that there is a multiplicity of ballooned hepatocytes. When their cytoplasm and nuclei are degraded and they are ballooned in size. I think you may also find the next slide of interest. This is an example of steatosis with the macrovesicular fat. This image is a reflection of our times in a certain sense, specifically current lifestyle of a significant portion of our society. This is Serotic Liver that contains, as I said, 30 to 40% of macrovesicular fat. BMI of that donor was 33, which is certainly elevated from an upper normal of which is 25. These characteristics would be, easier to accept or to take, except this is the liver of 11 years old child. And you can hardly think that this is an advantage to have 30% of your liver replaced by fat at this early age. The last image in this group illustrates the other end of the spectrum of the fatty liver disease, which is liver cirrhosis. Fatty liver disease progresses from normal to steatosis, to steatohepatitis, with or without fibrosis, to cirrhosis, and in few percent of cases to hepatocellular carcinoma. To conclude presenting different stages of fatty liver disease from the biobank, I'd like to share with you this example of cirrhotic liver. This is our donor, 525, 56-year-old Caucasian female. Content of fat is about 5% or less. BMI is 23.1, which is, all things considered, a very nice BMI indicated good amount of fat in the body. And of course, the slide has been stained for collagen fibers with Mason's trichrome. You see thick bands of connecting tissue that distorted normal architecture of the liver and therefore organs function. The fibrosis score in this stage is 4. This is the highest that can be given. And we can disclose that heavy alcohol consumption led to these conditions. Different stages of fatty liver disease I showed you on this and other slides are the basis of the tissue microarrays we offer. Microarrays are fragments of tissues organized on a glass slide in a way that can facilitate study of a specific stage of fatty liver disease, in our case, or other liver conditions or specific populations. And in our microarrays, they are of 3 millimeters in diameter to allow sufficient representation of the tissue for proper analysis. They are cut 4 micrometer thick and arrayed on a glass slides. We prepared slides for non-alcoholic steatohepatitis, steatohepatitis with history of alcohol use, steatosis without alcohol, and steatosis with history of alcohol, fibrosis of different backgrounds, non-insulin-dependent diabetes mellitus, and insulin-dependent diabetes. We also have additional control slides in form of a normal adult for baseline studies or characteristics. As well, we have a normal and disease pediatric array. To prevent tissue degradation, arrays are packaged individually in oxygen-depleted atmosphere at 4 degrees. This is an image of liver fibrosis microarray. They are provided unstained, but you can view stained examples on our website or data sheets, and you can do it adjusting magnification for better appreciation of specific histological features. Liver fibrosis array has 30 samples arranged left to right, top to bottom. Samples A1 to A4 has highest grade of fibrosis, fibrosis stage 4. They are followed by examples of fibrosis stages 3 to 0. Samples without fibrosis can have steatosis. This can be observed, for example, on sample E6. Finally, I'd like to illustrate to you one example of research facilitated by the arrays: analysis of activation of liver stellate cells. Smooth muscle actin is marker of activated stellate cells. On the left-hand side of the slide, you have the image of a periportal area, and in red, smooth muscle actin is stained. You see presence of this marker in the venules, arterioles, and bile duct cells. This is a normal location of smooth muscle actin in the human liver. On the right-hand side of the image, we see inside of the lobule, and we do not find cells expressing this protein in this area. This is a normal control that smooth muscle actin is present only in a specific structures, such as those present in a periportal region or sometimes in a mechanical support structures in the liver. From the same donor and the same slide, we have another view of the area away from these structures that normally express smooth muscle actin. And again, this is an area that is negative for this particular marker. But in contrast, in other specimens of the array, multiple cells expressing the marker are observed. And these are small cells which contain red dot, usually located or visualized in this case close to the nuclei. Stellate cells are relatively small as in comparison to hepatocytes. Therefore, the location of a smooth muscle actin appears to be overlapping with the nucleus. So the cells with the small red dot are activated stellate cells. Hepatic stellate cells are found in a space of distal, which is a small area between the sinusoids and hepatocytes. And the stellate cells is a major cell type involved in a liver fibrosis process, which is formation of a scar tissue in response to liver damage, as we saw on an example of a Serotic Liver slide. Use of an array allows analysis of a smooth muscle actin marker in a specific cell type and its location that is not visible in isolated cells or fractions. We also have another great example of the use of the array to study extracellular matrix. In companion presentation, we will hear about the relationship between alcohol consumption and accumulation of hyaluronan in the human liver. Dr. Michelle Pritchard of Liver Center, Kansas University Medical Center, will tell us about her research on this subject and applications of tissue microarrays. In conclusion, I would like to thank you, my colleagues whose work make the biobank possible. I also would like to encourage you to ask us any questions about scientific content of this presentation, as well as details of the biobank liver collection. Thank you all for your attention and stay safe.